For the nucleotide synthesis, what all do you want us to know about that again?
So for nucleotide synthesis, all I'm really concerned with here is that you know that our nucleotides are mostly made from amino acids. So we think about the carbons in these. So I gave you the whole pathway and we looked at it, but really let's look at this. Let me get back up to 33%. All right, 33 is the magic number, I think, to not have stuff go off to the side. But we can see how it goes going forward. So if we look at this, this is our purine, and almost all of these atoms, every atom in color in the purines here is going to come from an amino acid. Where does the sugar come from?
The pentose phosphate pathway.
Pentose phosphate pathway, I want you to know that, right? The ribose 5-phosphate. And then we also could have some that are going to come from, here's our formal tetrahydrate. So almost all the atoms. Here we have some bicarbonate, we'll get some, and also a formal tetrahydrofolate. Okay. There'll be 10 formal tetrahydrofolate, I believe. So mostly all amino acids, no pentose phosphate pathway. That's good enough. And then the same sort of thing for our pyrimidines down here. What else do you guys want to ask? Yeah, Patty.
There's a lot of the study that I read on one of the exams you posted about lipoproteins. Do we have to know the information to that level or just what we went over in class?
Just what we went over in class, but what?
Because it was talking about like a lot of the specifics of what they're exactly made out of,
Mm-hmm.
and to like the ratios, which we didn't really talk about.
So actually, in one of those exams, I'd actually ask what the percentages were, or did I ask for trends?
Like trends and what each one is.
So what is the trend for protein? So actually, first off, let's go ahead and what are our 4 lipoproteins?
The LDH is like very low density lipoprotein, low density and high density.
Okay, and then we have chylomicrons up here, right?
Those little ones.
Talk to me about the composition of these things. If you were to go ahead and talk about protein for them, what would it look like?
Isn't it like there's the least amount of protein in the very low density and then the highest amount in the highest density?
Right, so protein increases in this direction.
Yeah.
Okay. You need to know actually the roles of these things, right? So I could ask you some relative questions. So if we were to look at this and talk about, let's talk about lipids, right? The amount of triacylglycerols. Which of these, so the chylomicrons are kind of in terms of how we talked about roles. I would draw the same sorts of arrows and I would include the chylomicrons, but really in terms of roles, and we talked about, we didn't really describe them. Okay, so let's just go VLDL, LDL, and HDL. Where are my triacylglycerols going to be the highest? VLDL, right? So it's going to look like this. Okay, and the way I would reason that from what we've talked about is we've talked about VLDLs that mostly their job is they go out, they got triacylglycerols and cholesterol in them, right? They're dumping off, they're giving up their triacylglycerols to tissues, and at some point they've given up so much of those that we could change what we call them LDL. Okay. What's causing, when we talk about the density here, very low density lipoproteins, low density lipoproteins, high density lipoproteins, what causes these things to be more dense?
The lack of increase of the triacylglycerol.
It's more protein. They become more dense the more protein content they have. Okay. And by nature, the less lipids they have, right? So our triacylglycerols are going to go up as we go up here. They give up, they become LDL. What's the role of the HDL? It collects not triacylglycerols, extra cholesterol. Right. Okay. This is going to collect cholesterol and bring it where? Yep. Our LDL, where do they come from? They're VLDL that has given up enough triacylglycerols that it becomes, there's some density limit, some density threshold that somebody picked, okay? And maybe it moved around a little bit, but it's at a certain point right now. We're not even going to worry about what the percentages are based on what I covered in class. Where does HDL come from? High density lipoprotein. It's made in the liver to go out and sweep up cholesterol and then come back and deliver it back to the liver to either be used or broken down. Okay. What are the chylomicron? Where do they get made? In the intestines. And what do they do?
They allow you to bring the cholesterol into the liver.
They're going to bring the cholesterol and what to the liver? Triacylglycerols. Okay. And then they get absorbed by the liver. The liver then rebuilds the LDL out of the parts, let's say. Does that make sense what the roles are? So VLDL is going to take stuff from your intestines, your triacylglycerols, and your cholesterols from your intestines. It's going to deliver it to the liver. The liver takes it up, breaks it down, it reconstructs and makes VLDL, which are different lipoproteins, okay, although they may have a similar content to what chylomicrons have. They're going to then, the VLDL is going to run around in your blood, and it's going to be shedding off triacylglycerols until it's mostly proteins and cholesterol. It's then going to be absorbed into cells. The LDL actually gets fully absorbed into cells. It gets broken down, and they use that cholesterol. And then part of a separate process, the liver makes HDL to go around the body and sweep up extra cholesterol so it can take it back and degrade it, use it, apply it someplace else. Yes?
The HDL does nothing with triacylglycerols, just cholesterol.
HDL does nothing with triacylglycerols. It may contain a little bit of them but really it's not picking up extra fats.
So when you say reconstructing VLDL?
I'm sorry?
Reconstruct VLDL?
What? I did the first part of that sentence.
Sorry, like we talked about, you're taking cholesterol and triacylglycerols from the intestines to the liver, and
Yes.
you're reconstructing VLDL.
You're reconstructing a VLDL. Those are lipoproteins. Let's go look at a picture of one of those. So we talk about these things, and they get talked about as cholesterol, right? We talk about high density HDL is a lipoprotein, but people say the HDL is the good cholesterol, LDL is the bad cholesterol. I want my picture of one of these. I'm so ambitious in each of these lectures. Never going to cover that much. All right, so let's see. This is going to be the right one. Here we go. This is a lipoprotein. I don't know which one. Okay, probably not an HDL, and probably not really much of an LDL since it has a bunch of lipoproteins. Maybe it's a chylomicron. I don't know. But a lipoprotein is made up of these proteins. Let's kind of zoom out a little bit. A lipoprotein it's a complex of molecules, right? It's a macromolecular structure, and we have proteins in it. We have phospholipids around it, and then in the inside, and those are the phospholipids and the proteins are about transporting the cargo that's inside. The cargo inside are these triacylglycerols and cholesterol esters. And cholesterol ester is just cholesterol, except you have that OH on cholesterol, and that's just got a acetyl group attached to it, so it's an ester and makes it even less polar. Yes.
Do you even know that general structure of a lipid?
You need to know what the 4 parts are. 5 parts. Yeah, 4 or 5 parts.
So like the ones that are pointed out there?
Yes, those parts exactly. And the cholesterol on the outside, yeah, the cholesterol on the outside is also cholesterol. We talk about it as being a membrane lipid. Yes.
So
So the LDL is made in the liver and it drops off triacylglycerols, and then
Yes.
that turns it into LDL.
Yeah, so as you deplete those triacylglycerols, it becomes more and more dense. And at some point, it crosses a threshold and gets called LDL.
And then once it's in LDL form, then it's ignored into the cells for just to drop off more.
Drop off the cholesterol.
Okay, drop off.
It's mostly not been shedding cholesterol.
Okay, so
It's been shedding the triacylglycerols.
sheds triacylglycerols, turns into LDL.
Yes.
Then drops off.
Then gets absorbed.
Into the cell.
Gets absorbed and degraded, and the whole thing goes away.
Okay.
And then the liver makes these HDL. HDL is a separate lipoprotein that's job is to run around the blood and sweep up, collect extra cholesterol from cells.
Okay. Thank you.
Yes, Kendall.
So on your study for the exam,
Mhm.
you were talking about knowing like 4 amino acid synthesis.
Yeah.
And you're giving us like half of it on
I-
the study on the exam. So
I'm giving you half of it. What do you mean by that?
the beginning of it, the reactants.
I'm giving you the reactants. Yeah,
So
the reactants you're allowed.
Oh, okay.
Does that answer it? So for the quiz, you were allowed certain reactants. And a lot of people had memorized this. I was trying to show I was trying to show a transaminase reaction. And I was trying to do it not from, it was to glutamate or to alpha-ketoglutarate, not from alpha-ketoglutarate. And some people were making things starting with the valine. Valine is not one of the things you're given. Does that make sense? So the things you're given are what you were given on that quiz. Yeah. Well, let's hold on that because a lot of people have problems with that on the quiz. Do you want to see examples? So when I think about that, let me do it anyway, whether you ask or not. Okay. Because people did have problems on that. Let me give myself a little more space here. Okay. So reactions that were given, there are 2 reactions you have that I've said, hey, you got to memorize these reactions. Okay. And those one of those 2 reactions is helpful here. All right, let's my glasses. So what we've got is, I've got to do 4 reactions. Okay, 3 of them you pretty much are given, and the other ones you're going to have to know. So the first one that you have to know how to take ammonia out of the blood, right, and make glutamine. Okay, so we have ammonia, and you're given glutamate. You can't make glutamate as one of your 3, or one of your 4, or 2, or however many I ask for in the end. It could be 4. So I've got ammonia, right? And I've got that, and I'm going to use some ATP in this reaction. And then I've got glutamate. I really don't care which protonation states you use here, unless if I say I care, then you need to worry about it, right? But that was really more first semester that I cared which protonation state at what pH. It may come up again at some point, but nothing's jumping to mind for me right now. Okay, so I got this reaction here. It's probably reversible, but I have to look up the actual thermodynamics of it. So we're going to make ATP and phosphate, and we're going to attach this ammonia here to the glutamate. We're going to make glutamine. Glutamine synthetase, because it uses ATP. Okay, glutamine like that. All right, and so yeah, our enzyme here is glutamine synthetase. All right, so that's one of them right there. You have to know it already. Because this is part of that set of reactions where the next reaction would be to react that glutamine with alpha-ketoglutarate to make a glutamate, right? But just for this problem, I'm going to pass on that for now. Make sure you do need to know that reaction. Okay, so, go ahead.
Like when we put the ammonia on the glutamine, where does the oxygen go? Like, what happens?
When you where does the oxygen that was here go?
Yeah.
it leaves with the phosphate. Yeah, so if we were to draw it out, the phosphate's in here being added as a leaving group, and so that's where it's going to be. It's a good question. All right, so there's one. I've got two transaminase reactions I can make, right? I'm allowed glutamate, so I can transaminate pyruvate and I can transaminate oxaloacetate. So I'm going to go ahead and draw glutamate twice over here.
Do we need to show the top one if we're going to use glutamate?
You're given glutamate in these problems, so you don't need to. If you were going to use glutamine, so you've got one last one you find. If you need glutamine for one of your reactions, you would need to make glutamine.
Okay, got it.
Okay, but you could say, hey, I made glutamine up here, so here's my next one. Okay, and so this is a transaminase reaction. I'm going to react this with pyruvate. And I'm going to call that a transaminase. Well, no, I'm going to write the full word out because I'm not going to accept you doing TA on my quiz or exam. So transaminase. Okay, and then, so I'm going to transfer this amine from there over, well, draw it more carefully, from there to there. That's what a transaminase does. It's very specific for that. So now my glutamate becomes alpha ketoglutarate, which is one of my citric acid cycle intermediates. It doesn't matter for the problem because it's telling you what I can start with, right? And I'm not even being consistent with my protonation state between my things here, and I don't care. All right, so there's two, right? Pyruvate, you already know pyruvate is part of the glycolysis. 3, oxaloacetate, right? That's a citric acid cycle intermediate. So I'm going to go ahead and react glutamate again. With oxaloacetate. And what I really encourage you to do here in terms of structures is not memorize them all. I've got a subset memorized and I make them from other things. So these transamination reactions, I don't even know what the structure is for oxaloacetate. I don't have it memorized. I don't have the alpha ketoglutarate memorized. I do have my 20 amino acids memorized. And so I can make them from that. I know I just need to pull the amine off. I realize that may not be as helpful as I'm trying to make it sound because you may not have all your amino acids memorized. But glutamate and aspartate are good amino acids to memorize because they're some of the charged ones, right? Okay, and so there's my alpha ketoglutarate, and then I'm going to make aspartate now. Three. Okay? And then the fourth one is one you find in the book that you like to memorize. Okay, can I make fun of my lacrosse team members? And if you're on the lacrosse team, you picked a really complicated one. But I'm not going to fault you because you did it correctly. Okay, so the easy one here, there's one you can do in one step. I now have aspartate. I've now got aspartate, okay? Actually, I think I'm not going to even tell you which one this is, but there's one you can do with aspartate now that you've made aspartate. That's one step, okay? And it will be, it is in your book.
Okay, so wait, when you do, I know you said this, but so when you use each stuff, you have to have already made what we want to use.
Yes, but for these first three you don't actually have to make any of this, right? Because you're given everything for the first three, and the next one if you wanted to do aspartate to something else, you'd have to have made aspartate in an earlier one. You could just say, hey, I'll take this from here.
Okay.
And I'm not telling you guys to change.
Yeah. It thought it seemed a little complicated.
Morgan.
If you were given a question and you had to do this, are you going to, like on the practice, you gave the reactants and you did the class, or you
Oh, for transamination reactions? Yeah, I could do that.
okay.
So in any of these transamination reactions, I'd give you transamination reactions for anything, but I'm probably going to have you transfer it to or from glutamate because that's the way it happens, right? So I could give you, you know, well, I'd used valine before, right? But let's not do that. Let's use serine. People use serine, though. Did I use serine as an example? We use cysteine. Cysteine. All right. So it could be this is your reaction. Okay. So you've got this plus.
It's just like a random.
Well, it's not random. It's an actual amino acid.
Well, I
But
understand, but like we didn't mix there, like we didn't mix cysteine, so the range is.
yeah, in fact, I lost a carbon, didn't I? Well, I lost a carbon. Now let's do it like that. Okay. But that's not yet, but it's gonna be. But we could react that with glutamate. This is it's a transaminase. This is the transaminase. So. Mhm.
Is that one also
The one to make the aspartate, that's a transaminase.
Yeah, aspartate.
This is also a transaminase. Right, so a synthetase will use ATP. A synthase does not use ATP. The first one is a synthetase because it uses ATP.
Yes, but the one that we didn't finish, that one is.
I'm not going to tell you.
No, don't worry about this.
Are you transferring it to me? No. Are you transferring it to me? You're transferring it
But
to
you
me.
still use ATP.
We're still transferring it.
No. You're talking about this one that I didn't finish.
I'm talking about that.
Well, look that one up.
Right, Adam.
If that one doesn't use ATP, if it uses ATP, it's a synthetase.
Okay.
And if it doesn't use ATP, it's a synthase.
Could you go back up?
It's a transaminase.
Okay, that's
Hmm?
not a transaminase.
These are transaminases. The one in yellow, I'm telling you to go, if you want that one,
Yes.
go look it up.
But is it not gonna be a transaminase or?
It is not going to be a transaminase.
Oh, it's not?
No, if it was a transaminase, it would be super easy.
Oh,
Right?
Right.
It still isn't very hard. Right? Transaminases do these specific transfers, right? But the only two I can think of, maybe unless you guys have found one I haven't found, the only two that come from things in the citric acid cycle, right? If I'm not allowed to do glutamate, I can make alanine and I can make aspartate because I've got oxalic acid and pyruvate. I'm pretty sure I've not missed another one that I can just add an amine to and it becomes an amino acid, another intermediate. Okay, so down here I was doing another transaminase reaction. I'll finish it, even though I feel like I'm losing you here at this point. Okay, plus, and then now I've transferred this amine from glutamate over to there. It's a transaminase. And this is a generic transaminase reaction. So like on the quiz at the bottom, I said complete the reaction. It was a transaminase reaction. Okay. But this would not be one of my four because this was not one of the things I was given to start with, right?
So What is that?
That's alpha keto gonna be cysteine.
Alpha keto something turns into cysteine.
It's an alpha keto amino acid, and it's the alpha keto precursor that would become cysteine. I don't know what the exact name is.
This is like your last question based.
Yes. Yes.
Well, I don't even know why.
Okay. Bridget.
Can I ask a question, John, about that?
Yes.
Cool. So in chapter 18, I think.
18. Okay.
No, it's
Is it
17.
okay?
The first thing is on the study guide is like talking about biosalts. We didn't really discuss those in depth. Do you want us to know them?
What do biosalts do?
They help with digestion, like surfactants, right?
Right. They're surfactants in digestion. So I know we talked about them first semester, and I can't remember if I would have briefly talked about them when we were talking about we introduced fatty acids, unless I forgot. Do you?
They help, but, yeah.
You're going to get lipids. They're going to make you're going to get your lipids resuspended.
Yeah.
The lipids in terms of your diet. But let's just see if I talked about them at all. Because if I didn't,
I don't have it in my notes.
Yeah. I just have biosalts help.
I feel like this was really stupid. I said cholesterol.
it was semester one, and it often comes into semester two. But I need to do a better job of finding stuff faster.
From the liver to the intestinal lining. I have an error.
Did you draw that?
Okay, so here's where I should have gotten this earlier. Here's where we looked at, we're talking about the beginning of beta oxidation. Yeah, I didn't talk about, I know I didn't talk about it when I talked about LDL, so don't worry about biosalts. It's first semester, but
In the exam too from 2023, there was, it talks about malonyl-CoA. We talked about that.
malonyl-CoA?
FAS.
Yeah, malonyl-CoA is in fatty acid synthesis.
Do you want us to know the synthesis of malonyl-CoA? Because we want to learn.
We did go over it, right? Or we just talked about how much ATP it took.
It takes one ATP equivalent to make pharmaceutical
Oh, okay.
I think this was like really.
Or like what we need to know.
Let me see what I did, and so I'm gonna restrict myself to what I talked about here. So we'd gone over it before we talked about ketone bodies. Must be pre to this one. Oh, it's gonna be the one with all the tuberculosis stuff on it. All right, so here we are. So it's right here because we talked about how biotin was related, right?
Mm-hmm.
So here's how we're gonna make our malonyl-CoA. Okay.
Do you want us to describe?
I mostly want you to know that it's not. I mostly want you to know that it costs an ATP, right? You should always be able to tell me that the ATP, the role of the ATP is that it's providing a leaving group, okay? And then this is where we start talking about biotin. So you should be able to recognize biotin, to say that biotin's involved here. You should recognize biotin as a cofactor and be able to draw, whether it's loaded or unloaded, and draw the other form. Yes?
Can you talk about the cofactors from this? So it's biotin, there's the one from the quiz.
There's biotin, there's our tetrahydrofolates,
Yeah.
right? What else do we have?
The acetoacetate and the hydrobutyl hydroxy.
Those are not cofactors, but those are ketone bodies.
But we need to know that.
You don't need to know the structure. For those two, you just need to recognize, hey, those are ketone bodies, and talk about what their role is. Okay, and the role of ketone bodies is when you run out of carbohydrates, these are things that your liver makes from acetyl-CoA, right? You can get acetyl-CoA from fats, and it ships them out in the blood so they can go to your brain and other tissues so they actually have fuel. So it's the backup fuel when you don't have any carbohydrates. Yes?
On the exam, it was talking about lab. Is that something you're gonna?
Oh, that there might be something from lab? I feel...
Please don't.
I mean,
Please
I'm not
don't.
in
Please
lab,
don't.
so.
Yeah, that's true. That's not fair.
That doesn't. You made your choices, but
Okay.
I can't restrict what I ask based on somebody not being in lab. I haven't graded a set of lab reports yet, so I guess I'm going to not do that. But I am conflicted on that. I feel like I should put more things on lab because I know that people are. I haven't read your lab reports, but I know at least some of you are using ChatGPT to answer lab questions. I just know it's happening. Okay, based on what I saw last semester. So when that happens, it's not the analysis of these lab questions I'm asking is not going through your brain. It's going through ChatGPT. But I won't ask it this exam.
Thank you.
But I will ask it. I will ask it the next exam. Okay. Other questions? Yeah.
I don't think I wrote down in my notes as detailed as I wanted to for the why hexokinase is higher in the liver versus glucokinase.
Okay.
Can we just briefly talk about that again?
Sure.
Those are
This was last lecture.
Mm-hmm.
yeah. So kind of
That's what.
So let's talk about what we're talking about here is when we talk about higher, what we're talking about is the activity of these enzymes and whether these enzymes respond to the concentration, whether they change their rates based on the concentrations of glucose around. Okay, so we think about what the liver's role is. The liver is supposed to maintain glucose concentrations outside the cell and inside, or for the body, right? It's supposed to maintain glucose concentrations in this band, right? If glucose drops too low in your blood, you actually pass out, okay? Assuming we're not starving and we've adjusted with ketone bodies. But if glucose drops too low in your blood, you pass out. If it's too high, that's diabetes, and you have problems with that. So liver's going to maintain your concentrations here, and so the liver has to be able to release glycogen supplies if glucose is too low, and take up glucose if it's too high. So what you want is, as this is glycolysis, right?
Mm-hmm.
Okay, so if glucose is too, let's say, if glucose is too low, are you going to be converting it to glucose 6-phosphate? Do you want to if you're the liver?
No.
Glucose is too low, you want to break down glycogen and drop and kick it out into the blood.
Mm-hmm.
Okay, so you do not want glucokinase to be grabbing glucose and turning it into glucose 6-phosphate. You want to have glucose, right? And remember, from glycolysis, I don't think I have the pathway close, so I'm not going to go off of it. But glycolysis, that first step, glucose 6-phosphate to glucose is not the same reversible step for the other reaction. So these enzymes do not catalyze their reactions both directions because they're single arrow direction reactions. Does that make sense? Okay.
So the muscle's always gonna wanna do that.
But the muscle is never taking glucose and shipping it out to the blood. And all your glucose transporters are unidirectional, or they're not unidirectional. They all take no energy, which means they're going to essentially take glucose. Glucose is going to flow down a concentration gradient, right? So it's efficient to have your glucose transporters, whether they're in a muscle or otherwise. This is a muscle cell, let's say, and this is your GLUT transporter. I don't know which one it is. Okay, there are a bunch of those, but these things don't take energy. They are passive transport, and so what you want from the muscle is you want glucose to passively transport in always, right? You don't want it going out. So the way you stop it from going out is as soon as you get glucose into the muscle, it'll take me too long to grow this. So as soon as you get glucose in the muscle, the glucose gets grabbed. The glucose hexokinase is super active, right? It's active. It's fully saturated with glucose. That concentration is much lower than what's in the blood. As soon as glucose gets in the muscle, you take that glucose, you make it into glucose 6-phosphate, now it's not leaving. Okay, and your muscles aren't going to have, they're not even going to have the enzymes to take this back to glucose.
Is it not leaving because of the concentration gradient, or is it not leaving because it physically can't get out?
It can't get out because it's now charged. It's not going to go, it doesn't fit in this receptor. It doesn't fit in this not receptor, but this transporter. Okay, because either way, glucose isn't going to be passing through the membrane. It isn't going to passively make its way through. It has to go through a transporter, and this transporter doesn't work for glucose 6-phosphate. And as far as I know, there isn't one that does. The liver, you also have GLUT transporters. And what the liver, when it needs to release glucose, it releases a bunch of glucose in the liver cell. And now, if you're releasing glucose in the liver cell, right, you just have a lot of glucose in here. Glucose takes a lot of glucose, glycogen, it breaks it down. It then takes it to the glucose 6-phosphate to glucose. And there's a lot of glucose here and not so much glucose here. And so overall, you end up with that, right? I draw one arrow out. When it's really, it's coming in and out. It's equilibrium, but it's going to go more out than in. Does that make sense?
Mm-hmm.
Yeah.
If you like gave us this graph, what kind of questions would we need to know the answer?
So I'd be looking for you to be able to reason. I can give you a graph. I could talk about, I could find some other system between muscles and liver based on their roles, right? And which enzyme is which. So here I asked you when I talked about it in class, which of these do you think is the liver and which do you think is the muscle? Okay. Does that help? It's not an exact question. This isn't a memorization sort of thing. It's a thinking about what the roles are of the tissue and what makes the most sense. But you have one enzyme here that responds to the concentration in the blood, and you have one enzyme that no matter what, when that glucose comes in, immediately phosphorylates it. It's running at full speed all the time. Yes?
So, like, hypothetically, you said there was, like, a person that had low blood glucose, which enzyme would be more active?
Sure. Which one would be?
Glucokinase.
Hexokinase would be more active at low glucose.
But, like,
Because right here, if I have low glucose, we're talking about different glucose. At low glucose, hexokinase is more active.
yeah, but, like, I guess,
Glucokinase is less active because you don't want this. This takes glucose and makes it in glucose 6-phosphate. It's the at low glucose blood glucose, the liver has to ship glucose out,
okay.
not
So
trap it.
It doesn't, okay.
Okay. Yes.
On the study guide, you talked about the effects of adrenaline and norepinephrine on the muscle and liver.
Yeah, I didn't talk about that so much. I have to explain it more if I was going to do that. So adrenaline is fight or flight, right? So if I tell you adrenaline is fight or flight, right, or you're running from a bear, what pathways do you need to turn on? This is a fair question, as long as I tell you you're running from the bear. Okay.
You would turn on glycolysis, like any pathway that allows you to get energy, and turn off like gluconeogenesis and glycogen synthesis and fatty acid synthesis.
Okay, you're running from a bear. You're saying stuff. I'm sorry. I want to get this down. So you're running from a bear.
I was kind of just talking.
No, it was good. Okay, you're running from a bear. Which pathways are turned on and which pathways are turned off?
Turn on glycolysis.
Right, so turned on. You're right. Whatever gives you energy, right? Glycolysis, beta oxidation. Okay. What else? Glycogen breakdown. I'd probably also specify the liver, because in the liver you have all these pathways possible.
Mm-hmm.
Certainly, and it's not the case in the muscles. What else? Citric acid cycle.
Oh, I think I answered this in class the other day.
Electron transport chain, right? Turned off,
Gluconeogenesis.
gluconeogenesis.
Fatty acid synthesis.
Glycogen synthesis.
Fatty acid synthesis. You said glycogen synthesis.
Would like nucleic acid synthesis even be relevant to this? Or is it separate?
Not really. It kind of, we haven't talked about it. It's not really related to energy. I mean, these are the ones we've talked about, right? So your body's not going to focus on growth sorts of things if you're running from a bear, but I wouldn't worry about those. Okay. In terms of, let's say you're running from a bear, and so what if you're also starving?
That you stop it, cancel out. You do like muscle, you break up the muscle.
Okay, going to be breaking down muscle, so
Keto.
you're going to be making,
Ketone.
okay, you're going to be making ketone bodies, right? Ketone body synthesis. Okay, this is the liver, right? You're obviously going to be breaking down ketone bodies in the tissues that receive it, but we're not going to worry about that. Ketone body synthesis, what else is going on in the liver? You've already alluded to why it's going on. I've already heard that part. How about the urea cycle? I mean, it's kind of always going, but you would turn it up because you're breaking down lots of proteins for fuel, right? And you could talk about amino acid breakdown. That isn't really a pathway for us, but it's a giant pile of pathways. Questions?
So we've done like beta oxidation, fatty acid synthesis, ketone.
For this group of
Yeah.
things. So it's been the 2 chapters and then a little, right? I don't know, is it 17 and 18?
17, 18, and
it's
17.
yeah. So it's the fats, so it's beta oxidation, fatty acid synthesis are the 2 main pathways you need to know. Then we have some amino acid stuff we've got, which is a very scattered set of pathways. So that's why it's, hey, give me 4 reactions, that sort of stuff. And we talk about those 2 major reactions that are going to capture ammonia. Yeah.
So for the list on the left, we're talking about, say it specifically is in the liver because the liver is interested in all the pathways.
Yes.
And it's starting ketone body synthesis specifically in the muscles.
No, that's this is, you're also starving. That's also liver.
That's also liver.
Yeah, this is all liver because I can turn things on and off in the liver. I have the opposite pathways for most things. If I was talking about the muscles, are muscles going to make ketone bodies?
No, they break down proteins.
They'll break down proteins and then they'll use the amino acids, right? They'll use the amino acids. They'll be either ketogenic or glucogenic, or sometimes they're both. Okay, and they're going to use the carbon scaffolds out of that. What else is going to happen in the liver? Certain muscles. What do muscles do when you're starving? Okay, so we're going to say break down amino acids. Great. Maybe what I'd say is, I could say, what do they do? What do they produce that they might not produce in large amounts otherwise? And give to the blood.
This one are also active.
What was that?
Are they also active in this scenario, or are they?
Doesn't matter.
Ketone bodies?
They're not going to make ketone bodies because they use ketone bodies. Making ketone bodies would be making fuel for somebody else, right? They're going to take if they have acetyl-CoA, they're just going to use it.
Are you talking about like lactate?
I'm talking about alanine. So yes, here it matters whether you're active or not. If you're active, then you'll be making lactate, right? But in terms of starving, why are you making alanine?
of alanine.
Doesn't it like
That's how you're getting, that's our alanine glucose cycle with the liver. Let's see, was it this one?
It's how we can get glucose to keep going.
So you've got, no, it's not about glucose to keep going, it's about you're breaking down amino acids. I mean, it's just giving, some of those give you glucose. But you've got all these amino acids being broken down. This is all the amino acids. Okay? And then they're going to hand off their amines to alpha-ketoglutamate glutamate, because glutamate's our intermediary of everything, right? But all your amino acids, they all have amines on them. You can't just make those into ammonia and dump them into the blood. It'll kill you. So what you need to do is you need to transfer those amines to pyruvate that then get to make alanine. Alanine's what goes to your liver, and then alanine will give its amines into the urea cycle through glutamate. Okay.
And what were you mentioning about the active versus non-active? Because I wrote in my notes that if it's active, we do alanine.
If we're talking about an active muscle, right? And you could be starving and active, right? But if we're talking active and starving, you're also going to be making lactate, okay? Because if you're really active, we are as animals not designed to take in enough oxygen to keep up.
So active muscles do lactic acid.
Are going to make lactic acid. So and there I would say, you're sprinting, you're starving and running from the bear, right? So you're sprinting hard. You're not going to be bringing enough oxygen. You're going to be making lactate, which will go into your liver that will regenerate it into glucose. But that's all about not having enough oxygen to fully oxidize it. Maddie.
Is alanine produced when you're starving regardless if the muscle is active or not active?
Alanine's going to be always produced, right? Because you're always turning over some muscle. But in terms of upregulating it, if you're starving, you're going to be breaking down muscle for fuel, and you're going to make your urea cycle is going to be a lot more active. Does that help? So you have to make a lot more alanine.
so both are up by you?
Both in terms of making lactate and making, what's both? Both the other ones lactate?
Alanine and lactate.
Yes.
Both operate in 100%.
If you're starving and active, right? Lactate's going to be about being active, sprint, weightlifting, something. Gardening, probably not, right? Because you don't make a bunch of lactic acid and then fall over and have to wait. Your muscles don't hurt, hopefully, and then for a while. Does that make sense?
Yes.
Okay. Did you?
Yeah, I just got a random question. Why is alcohol? What happens when alcohol is bad for your liver? So what's happening?
Oh. I mean, your liver is detoxifying it. But why is alcohol bad for your liver? I mean, I should know this. I don't. I don't know exactly what it's doing, right? It is a poison, but I don't know how it damages your liver.
Yeah.
Sorry. I'll try and look that one up. Yeah.
When you're fasting and running and you're turning on the urea cycle, is that because you can kind of connect it to the citric acid cycle and produce energy that way?
So when you're fasting, when you're starving and running, or fasting and running, either way, right? Yep.
Talks about ketone bodies, mixes, and urea cycle.
Right. So the urea cycle does actually, we didn't go over it, but it does actually generate a little bit of energy. But you're not. It's more about efficiencies. You're not doing it to make energy, if that makes sense.
Yeah.
The energy, the main point, the reason why the urea cycle is running is to take care of the amines. The main reason you have the amines when you're starving is because muscle's being broken down to make energy, if that makes sense. But it's in two separate tissues. Your muscles break down, right? You get the carbon scaffolds of those, and that's where most of your energy is, the amino acids. And the urea cycle runs to get rid of the ammonia. It is not running, it is not upregulated to produce energy, if that makes sense. It's not its role.
Okay. So the main point is urea cycle just gets rid of ammonia.
It's getting rid of ammonia. It's otherwise you'd have ammonia. It's to take in those amines.
Okay.
Ammonia is not made, really. You're avoiding that.
Okay.
Any other questions?
I have one question about biotin.
Okay.
The structure of it.
Yep.
So we have to list the structure.
Yep.
The loaded and unloaded.
Yep.
Where is the loaded and unloaded?
It is where we talked about malonyl CoA. It's right here.
Yeah.
So this is. Do you want to know which lecture this is?
This was from 223.
20.
223.
220 is what I've got here, but maybe.
Yeah.
But you've got this particular. You've got this right here on 223. You said.
Yeah.
Okay, sure.
So we just talked about like if you have an oxygen attached to the carbon.
So if you want to know where biotin is loaded, this is loaded biotin.
On the nitrogen.
This is unloaded and that's unloaded. Yes, you're putting a carbon dioxide on there essentially. It's carboxylate, but
Okay, so there's nothing with like
the chain is just what attaches it to the enzyme. It's like our lipoate. That is attached to a lysine. This is probably a lysine. Okay. All right. If you have more questions, feel free to please reach out and let me know.